Bismillahirrahmanirrahim. Now, today I want to discuss about one of the gram-negative organism, although it lies in gram-negative organism. But this organism which I want to discuss is very important in the pathogenesis of the different diseases of the gastrointestinal tract. Now this is which is called as the Helicobacter pylori. At one time it was called Campylobacter pylori. It was considered as commensal in the stomach and other parts of the small intestine. But nowadays this is the hot topic of the 21st century. I am Professor Sayyid Ali Hazar. I am honorary global professor of medicine without any fame in economics, and I am addressing to every student, whether appearing in the third year, fourth year, final year MBBS, or appearing in the membership examinations, or in the Pakistan, or the fellowship examination in Pakistan, as well as those who are appearing in the foreign examinations of the European or the American territories. Now, this organism is important to understand from every angle. You should know. The thing is that it is a very important pathogenic agent. I already said it. The important part of this organism is that it has been proved it is the causative agent of many of the diseases which were at one time related to the acid and the mucin in the stomach at one time. And that is the pathogenesis of the acid peptic disease. And those people who are suffering from the acid peptic disease as well as GERD, or in certain cases not as a whole, as you can say, in the minority of the cases, it may be associated with some of the conditions of the large intestines also. So this organism is important in this context. Every part of or everything which is available in the literature should be discussed. Should be discussed because if you are appearing in the examination, and there are practical examinations specifically speaking, the examiner put you any question in front of you. Be ready for it. Because I am also the examiner, or rather senior examiner of the fellowship examination in Pakistan in internal medicine, and some of the membership examinations of the College of Physician Surgeons. The thing is that this organism should be known broadly rather than going very quickly about it. So this is what is the importance of this organism. Now, reduction in importance is this, that the Helicobacter pylori, I said that it is also known as the Campylobacter pylori, is a gram-negative microaerophilic, means only the thing is that the small oxygenation is needed. Microaerophilic, it is spiral helical bacteria, spiral-shaped bacteria, like a spiral sheet. Like a parachute is a big organ, big bacteria. It is a small bacteria, usually found in the stomach. It's helical in shape, from which the genus name Helicobacter derives. Heliculation. It is thought to have evolved in order to penetrate the mucous membrane of the stomach and thereby establish infection. Now, this bacteria was first identified in 1982. It is the graduation of myself is concerned, 1982, from the Dow Medical College. At that time, it was described, identified by Australian doctors Barry, Marshall, and Robin Warren. And these are the people who gave us a new concept about the pathogenesis, or you can say the primary etiology of any disease. Rather than the stomach, only it may be possibility that those diseases what seems to us at one time that it is not related with any microbial agent. Now every disease is now under research, that probably they are the what is called initiators of the problem. So this was the first one, Helicobacter pylori by Barry Marshall and Robin Warren. Now they have given this concept in 1982, and they wait for the in America, because America has got a lot of the research facilities there. They proved in America in 1990 that they are right in the sense that they are the initiators of the problems, and they can give rise to specific illnesses of the stomach as well as other parts of the intestine. And then they gave the Nobel Prize of the medicine in 1992. So after 10 years, they got this Nobel Prize. After much research has been done in America and Canada, and they declared it that they are correct. Now, Helicobacter pylori has been associated with the cancer, adenocarcinoma, and of the malt that is called mucosa-associated lymphoid tissue in the stomach. It is related with it. Esophagus, colon, rectum, or tissues around the eye even, they are important. Termed the extranodal marginal zone B lymphoma. So it is associated with the initiator of some types of the B cell lymphoma of the sighted organ, and of the lymphoid tissue in the stomach, termed the diffuse large B cell lymphoma. So they are associated with the acid peptic disease in one way, the adenocarcinoma, the malt, the extranodal marginal zone B cell lymphomas, and at any part, the esophagus, colon, rectum tissues, even the eyes, and also the diffuse large B cell lymphoma of the stomach. This should be remembered. Associations, predisposition of this organism. In the examination, we asked about it, and they said they are, of course, they are the initiator of the predisposing factors of the acid peptic disease, but they don't know about that there are many other tissues are also involving. So that is so important. That is why I have given this lecture in at least 2 parts, because it is a long discussion. I don't want to involve you in the big discussions, but for the sake of gaining much knowledge about this organism, you have to listen to me. So I have divided this discussion into 2 parts. Otherwise, I can go through. Because at one time, when you will go for the acid peptic disease, which is an important disease, definitely, about 20 to 30% of the cases in my OPD is related with the acid peptic disease. That is why it is so important. The Helicobacter pylori infection usually has no symptoms, but sometimes causes the gastritis. So it is a part of the chronic gastritis. To add the list, which as previously mentioned, that it is the cause of the chronic gastritis, specifically speaking the B, gastritis B, which is the gastritis of the old age. In the young age is no problem, but in the old age this is seen growing much and much. And they said about 60 to 70% of the world population of the geriatric group, they are growing the Helicobacter pylori in their stomachs. So this disease will cause the chronic gastritis B, and it can cause the ulcers of the stomach, that is the ulcerative type of the stomach the disease, that is the acid peptic disease or ulcerative peptic disease, or even the duodenum is involved. This is part of the first part of the duodenum, definitely involved. The infection is also associated with the development of certain cancers, that is mold, the B-cell lymphoma, and the large cell lymphoma of the stomach, and in addition to the adenocarcinoma. Many investigators have suggested that the Helicobacter pylori causes or prevents a wide range of other diseases. It can cause, it can prevent, because it is the usual war which is going on as a coliform bacteria in the intestine. If you suppress it, then the other disease will appear. That is why it prevents a wide range of other diseases. But many of these relationships remain controversial. Research is going on whether they are involving it or not. For example, if you give the antibiotics and you are killing the coliform population in the small intestine, it can give rise to what is disease which is called the antibiotic associated diarrhea, or also called the Clostridium difficile associated diarrhea. Just like this. If you kill it 100%, then because it has also the preventive role, so that preventive role will be gone. The suppression of the H. pylori is needed, that's why. Only not the 100% eradication. So this discussion is going on in the world, in the different seminars. Research papers are coming day by day for this organism. Some studies suggest that the Helicobacter pylori plays an important role in the natural stomach ecology. Ecology, you know that the milieu interior of the stomach, where the functions of the stomach is going place. For example, by influencing the type of the bacteria that colonize the GI tract. That is why there is an important role in normal stomach ecology. Because there are other type of bacteria also present as commensals, as a part of the coliform bacteria. It is an important role in ecology of the stomach. Other studies suggest that the non-pathogenic strains of the H. pylori may beneficially normalize stomach acid secretion and regulate the appetite. On the other hand, it normalizes the pH of the stomach and to optimize for the optimization, and it can regulate the appetite. In 2015, it was estimated that over 50% of the world population overall had H. pylori in their upper GI tract, with this infection or colonization being more common in the developing countries because they are eating much and much, and they are drinking the beverages, and they are drinking the cold drinks, etc. And this type of the H. pylori is coming from the outside, and it helps in the development of the colony of the H. pylori in the stomach. This is the concept. In recent decades, however, the prevalence of the H. pylori colonization of the GI tract has declined in many countries. But in recent studies, because of the awareness, because of the physicians which are going on the correct path, they eradicate this infection to much extent, and that is why their prevalence of H. pylori associated with the diseases of the stomach has been declined. Now this is the microscopy of the organism in the tissues. Here you see this organism is a spiral, very small bacterium, spiral shaped, and here you will see they are aggregated here and this in this fashion. This one, look this one. It is small, spiral shaped bacteria, spiral shaped bacteria. This is all there. So the transmission of the H. pylori is this, that it is contagious although the extra route of the transmission is idiopathic. They don't know the which of the water, either water is involved or the alcohol is involved or other dietary factors are involved. They don't know exactly. But definitely the H. pylori which is coming in from the external sources will enhance the colonies of the H. pylori because we have it eradicated completely, 80 to 90%. It is negative for all tests for H. pylori bacteria, and after 6 months it recurs. So recurrence of this is unusual because you have killed it most of the part, and the patient was symptom free. And that is the question. That is exogenous source may be possible. Person-to-person transmission by either the oral kissing, mouth feeding, or fecal-oral route is the most likely. It is possibility like this. This is another route of the transmission of this infection, possibly. Consistent with these transmission routes, the bacteria have been isolated from the feces, cultured from the saliva, cultured from the dental plaque of some infected people. So it is also seen in the dental plaque, it is also seen in the conjunctiva. Findings suggest that the H. pylori is more easily transmitted by gastric mucus than saliva. It is more easily transmitted by gastric mucus than saliva. The saliva is definitely a part of it, but rather the gastric secretions of mucus are the more important. Transmission occurs mainly with families in developed nations, yet can also be acquired from the community in the developing countries. The developing countries following the developed countries. If they are using something, they are promoting the KFC or McDonald's, the franchised, franchisation of these companies will be developing in the developing countries. It may be the source of infection of this H. pylori. We don't know exactly because epidemiologists are working on it. H. pylori may also be transmitted orally by means of the fecal matter through the ingestion of the waste tainted water. So a hygienic environment could help decrease the risk of H. pylori infection. Hand washings and the food should be prepared at the home. Avoid the cold drinks, avoid the outside food may prevent this infection. It is possibility. So already mentioned out that it is gram-negative helix-shaped bacteria. It is 3 micrometer long with a diameter of 0.5 micrometer. It is a very small one. H. pylori can be demonstrated in tissue by specific stain, gram stain, or Giemsa stain, hematoxylin eosin stain, and rather the very specific stain is Warthin starry silver stain, and acridine orange stain, and phase contrast microscopy. You said in the examination that it is gram negative, yes, but Warthin starry silver staining is more specific. So you should answer like this. Although it is gram negative, but Warthin starry silver stain is more specific. And phase contrast microscopy, you can do it and can demonstrate it. It's capable of forming the biofilms and can convert from the spiral to a possibly viable but non-culturable coccoid forms can occur. It is rolled up like this, that the structure or the morphology of this organism very, very close to the cocci. Helicobacter pylori has 4 to 6 flagella. If there are flagella, its movement is very good. At some location, all gastric and the enterohepatic Helicobacter species are highly motile owing to the flagella. It has got 4 to 6 flagella. The characteristic sheath flagellar filaments of the Helicobacter pylori are composed of 2 copolymerized flagellins. The flagellins are FLA A and FLA B. This is important in the pathogenesis of the acid peptic disease as well as the adenocarcinoma of the stomach. Physiology is that the H. pylori is a microphoric organism. I said that it requires oxygen but very low concentration than in the atmosphere. So it contains a hydrogenase that can produce energy by oxidizing the molecular hydrogen H2 made by the intestinal bacteria, which consume it by like this and survive like this. It produces oxidase, it will produce the catalase and its urease. This is the most important enzyme secreted by the Helicobacter pylori. The Helicobacter pylori possesses 5 major out membrane protein families. The largest family includes known and putative adhesins, outer membrane in a plasma lemma outside the first layer of the cell wall. It has got the protein families. And one family is adhesins. The other 4 families are porins, iron transporters, flagellum-associated proteins, and proteins of unknown function. PUF, that is called protein of unknown function. Like other typical gram-negative bacteria, the outer membrane of H. pylori consists of phospholipids, liposaccharides, and they have got the O antigen of the lipopolysaccharide, may be fucosylated and mimic Lewis blood group antigens. It is very mimicking the blood group A or B, just like this, found on the gastric epithelium. They were called the secretors, and those people who have got the secretion contain the group A or group B, blood group A or blood group B. So they are called secretors. So it is very, very mimicking the O antigen of the lipopolysaccharide. You can differentiate between secretors and the non-secretors. The O antigen is a very important antigen. If it is negative, it means that it is not harmful. It becomes commensal, it means that. The outer membrane also contains the cholesterol glucosides, which are present in few other bacteria. It is a long, this is the physiology and biochemistry has been described with some part of the microbiology. Everything is described so much so that you know that it is the flagellum, it has got flagellum proteins, it has got the enzymes which have got the urease also, other than oxidase catalase. Please try to remember this. And they are secretors just like the lipopolysaccharides, and they mimic, you can identify them, because O antigen, if it is negative, it means that they are non-secretors, and non-secretors have got less chance of infection than the secretors. And this is the organism. Look, this organism is spiral shaped. These are flagella, 4. Flagella has got its own specific proteins. And here are the flagellas. And these flagellas, other than that, these are the microsomes containing the urease. And there are exotoxin also present on the outer membrane of the bacteria, circulating toxin. And then you can see the type 4 secretion system is operating in this bacteria. Enzymes which are called lipases and proteases and immunosenases other than urease. The urease is the main enzyme which should be addressed. So it is coming out from the host cell. The host cells are affected by all of this. They are called effectors, and they are actin modulating, remodeling occur, and the interleukin 8 induction, host cell growth, and apoptosis inhibition, because apoptosis is a very important part played in the stomach and in the lining of the other intestine. So it will go out, and the reproduction of the cells will occur. It will die, and new replacement occur. Now, at that point, if it is introduced, where there is a breakdown of the mucosa and regenerating point, the effective if it is introduced in this phase, it can cause the metaplasia in the stomach, and that is the pathogenesis of the adenocarcinoma or the lymphoid type of the carcinoma in the stomach. The genome of this Helicobacter pylori consists of a large diversity of strains, and hundreds of the genomes have been completely sequenced. The genome of the strain is 26695, consists of about 1.7 million base pairs. It's called diversity of strains. Genomes are there, therefore, with some 1576 genes. The research is so much so that they have described all this. The pangenome, that is a combined set of the 30 sequence strains, encodes 2239 protein families. They are the orthogonal groups, OGs. Among them, the 1248 OGs are conserved in all the 30 strains and represent the universal core. It is a universal core for them of the disease. The remaining 991 OGs, they have got the correspond to the accessory genome in which the 277 OGs are unique, that is, OGs present only one strain. So it is the least important among them. Now this is a proteome. It is a genome, it is a proteome. The proteins of the H. pylori have been systematically analyzed by the multiple studies. And more than 70% of the proteome have been detected by the mass spectrography, spectrometry, and other biochemical methods. In fact, about 50% of the proteome have been quantified. That is, we know how many copies of each protein are present in a typical cell. So the production of the protein, different parts of the cell of the bacteria is important in the pathogenesis. Furthermore, the interrectome of the H. pylori have been systematically studied, and more than 3,000 protein interactions have been identified. And these interactions are important in the production and pathogenesis of the disease again. And they later provide information how protein interact with each other, for example, in the stable protein complexes or in the more dynamic transient interactions. This in turn helps researchers to find out what the function of the uncharacterized protein is. For example, when an uncharacterized protein interacts with several proteins of the ribosome, it is likely also involved in the ribosome infection. Nevertheless, about a third of all, there are around 1,500 proteins in the H. pylori remain uncharacterized, and their function is largely unknown because there are different types of strains. H. pylori, each strain have got different type of functions. Around 1,500 proteins still are there in the assembly, which are not known what they are doing in the pathogenesis of the disease. The pathogenesis is a adaptation to the stomach. How they are survived in the presence of the high pH and in the acid environment. To avoid the acid environment of the interior of the stomach, lumen, the H. pylori uses its flagella to burrow into the mucous lining of the stomach to reach the epithelial cells underneath. In the first layer of the mucosa is aerated, and it reaches inside the epithelial cells, where it is less acidic. So it is one of the parts that is why the survival is possible. The H. pylori is able to sense the pH gradient. Just as you are detecting a pH of anything, it has got the ability to sense it by its receptors in the mucus and move towards the less acidic region, and that is called chemotaxis. From the higher acidic region to the lower acidic region, and that is called chemotaxis. This also keeps the bacteria from being swept away into the lumen with the bacteria's mucous environment, which is constantly moving from its site of creation at the epithelium to its dissolution at the lumen interface. So afterwards, when its survival is possible in this, now it is spreading, underneath with the help of the mucus which is flowing from the stomach to the other parts are involved of the stomach. In this fashion, it is spreading in the stomach. 1 or 3 or 4 or 5. If it penetrates like this, it can spread in the layer form because of the mucus movement. The diagram is showing how rich is the epithelium of the stomach. Now, this is the penetration, abrasion. It's embedded now in the first, lining of the stomach, that is epithelium. It is the columnar epithelium, it is going inside it, and here it can tolerate the acidic pH. That is why. Now, H. pylori is found in the mucus on the inner surface of the epithelium and occasionally inside the epithelial cells themselves. It adheres to the epithelial cells by producing adhesins. It adheres to the epithelial cell by the protein, the enzyme which are called adhesins, which bind to the lipids and the carbohydrates in the epithelial cell membrane. The defense is gone then for the stomach lining. One such adhesin like BABA bind to the Lewis B antigen displayed on the surface of the stomach epithelial cell. They are secretors for some nations. H. pylori adherence via BABA is acid sensitive and can be fully reversed by the decreased pH. It is fully reversed by the decrease. If you decrease the pH, it is worse, fully reversed. It has been proposed that the BABA's acid responsiveness enables the adherence while also allowing an effective escape from the unfavorable environment at the pH that is harmful to the organism. If you are lowering it much, then the organism cannot be survived. Another such adhesin, SABA, binds to the increased level of the Lewis antigen expressed on the gastric mucosa. Another research saying that this is another one, which adhesin is SABA, which is also involved in the pathogenesis of the disease. Now it has got the enzyme which is called urease, and it is how it is formed inside. It is inside the cell, and it is going out from it by the binding. Urease is formed just like in our cells. The enzymes are encoded in the microsomes, and microsomes are reaching here, and from the exostosis it will come out urease, and urease is producing, and it has got a very important pathogenetic effect on the disease. In addition to using the chemotaxis to avoid areas of low pH, also neutralizes H. pylori also neutralizes the acid in its environment by producing large amount of urease. Urease is a protective type of nature of enzyme. We break down the urea present in the stomach now, which is present in the urea, and form carbon dioxide and ammonia. The ammonia, and means that it alters the pH of the stomach. These react with a strong acid in the environment to produce a neutralized area now around the H. pylori. Now, urease knockout mutants are incapable of colonization. In fact, the urease expression is not only required for establishing the initial colonization, but also for maintaining the chronic infection. If it is present, it means that it's chronic gastritis B, or it is acid peptic disease. Simple as that. Adaptation to the high acidity pH also mentioned that it is the ammonia which is important substance which is releasing. Helicobacter pylori arginase, a bimetallic enzyme, binuclear amine-2 metalloenzyme or arginase, crucial for the pathogenesis of bacteria in the human stomach. A member of the urease hydrolase family catalyzes the conversion of all-arginine to all-orthonin and urea, where orthonin is further converted to the polyamines, which are essential to the various critical metabolic processes. So another important thing is that it will form ammonia, and ammonia will definitely causing the other pathogenic mechanisms. This provides the acid resistance now, and is thus important for the colonization of the bacterium of the azad ho gaya hai. Now it is becomes freely moving from one side of the stomach to the other side of the stomach, because now the pH is altered and it is saved. Arginase of the H. pylori also plays a role in evasion of the pathogen from the host immune system, mainly by the various proposed mechanisms. Again, the arginase competes with the host inducible nitric oxide synthetase for the common substrate arginine, and thus reduces the synthesis of the nitric oxide. Nitric oxide is an important component of the innate immunity. And this is from the innate immunity, and the nitric oxide, and an effective antimicrobial agent that is able to kill the invading pathogens directly. It is fighting with this nitric oxide. Alteration in the availability of the arginine and its metabolism into the polyamines contribute significantly to the dysregulation of the host immune response to the H. pylori infection. Inflammation, gastritis, and ulcer. It invades the lining of the stomach and the duodenum and other parts. So the ammonia produced to regulate the pH is toxic to the epithelial cells, as are the biochemicals produced by H. pylori, such as proteases regulating cytotoxin A. This damages the epithelial cells, disrupts the tight junctions, and causing the apoptosis. It is induced apoptosis because not natural cell death. It reduces the life of the lining of the stomach, and regeneration process. It needs now regenerative process very quickly, and it will not cope with this. And certain phospholipids also penetrating and damaging the stomach lining. Cytotoxin associated gene that is called CAGA can also cause inflammation. It's potentially a carcinogen. It is potentially a carcinogen. So this is a CAG-A can cause inflammation. Colonization of the stomach by the H. pylori can result in the chronic gastritis, B specifically, and inflammation of the stomach lining at the site of the infection. And H. pylori cysteine-rich proteins, particularly the A, are known to trigger an immune response causing inflammation. The H. pylori has been shown to increase the levels of the COX-2, that is cyclooxygenase inhibitor 2, in H. pylori positive gastritis. Again, this is harmful to the stomach lining. Chronic gastritis is likely to underlie H. pylori related diseases. So acute gastritis, it will cause the chronic gastritis, it will cause the acid peptic disease, that is the inflammation, gastritis, and in some parts it is the lining which is gone, causing the ulcerative type of a disease, the ulcer peptic disease. So these ulcers are found in the stomach and duodenum, result from the consequences of inflammation, allow stomach acid and digest the enzyme pepsin to overwhelm the mechanism that protects the stomach and duodenal mucous membrane. Because you know that the important thing is the acid, it is the fight between the acid and the mucus. The location of the colonization of H. pylori, which affects the location of the ulcer, depends on the acidity of the stomach. In people producing large amounts of the acid, H. pylori colonizes the pyloric antrum. Large amount exit to the duodenum now, and because of the relaxation of the pyloric sphincter, to avoid the acid secreting parietal cells and the fundus near the entrance to the stomach. Cells express relatively high levels of the prostaglandin L1 that protects these cells from the H. pylori induced immune destruction. In people producing normal or reduced amount of acid, H. pylori can also colonize the rest of the stomach. Where there is less, you are suppressing it day by day, then H. pylori chances of colonization are more. It means that those people who are taking the H receptor antagonists or PPI in a doses for a very prolonged period, they can cause the colonization of the H. pylori and cause these problems. And it can cause the metaplasia and causing the, again I'll say, the adenocarcinoma or lymphomas or malt, etc. So these responses are this inflammatory response caused by the bacteria colonizing near the pylorus antrum-induced G cells in the antrum to secrete the hormone gastrin. G cells around the pylorus causing the gastrin. What is gastrin? Now, gastrin will induce more production of the hydrochloric acid by the parietal cells. Gastrin stimulates the parietal cell to secrete now more acid into the stomach lumen, and it is more than the defense mechanism now, that is the mucus, and over time increases the number of the parietal cells as well. Hyperplasia occurs. It is reactive hyperplasia. More are the parietal cells, more are the hydrochloric acid. The increased acid load damages the duodenum, which will eventually result in the ulcers forming in the duodenum. That is the production of the duodenitis and causing the duodenal ulcer, and it will cause the acid peptic disease, which is involving also the duodenum. When H. pylori colonizes other areas of the stomach, the inflammatory response can result in the atrophy of the stomach lining and eventually ulcers in the stomach. This may also increase the risk of stomach cancer. So the day by day, there are chances it may be possibility of the formation of the adenocarcinoma. So this is one which is a very important thing, is the CAG pathogenesis island, CAG pathology island. And this description is given in the microbiology that say that the genes of H. pylori increase the genes of the CAG pathogenesis island, because this is the island which is in the stomach, in the other parts of the hollow organs. In the Western country, they researched it out, and they said that the type 4 secretion system expressed in this CAG pathogenesis island injects the inflammation-inducing agent that is called peptidoglycan from their own cell walls into the epithelial cells. And the injected peptidoglycan is recognized by the cytoplasmic pattern recognition receptors, the immune sensor that is, and stimulates the expression of cytokines that promote inflammation. The type 4 secretion apparatus also injects the CAG-PA1 encoded proteins CAG-A in epithelial cell when it disturbs the cytoskeleton of the stomach. It returns to the adjacent cells, intercellular signaling, cell polarity, and other cellular activity. That is why it's so important. So it is a CAG pathogenesis island. I think most of you doesn't know about this. This is the island actually. CAG. It enhances, H. pylori enhances the genes expressed by this island, which is present in the hollow organs. So this thing is new for you. Please try to remember if you can remember. Cancer is produced because if you are promoting you are enhancing the free radicals in the H. pylori, like for example the hydrogen ions and other ions, free radicals, and it can increase the rate of the host cell mutation. The mutated cells are formed in the stomach lining, and if they are mutated, it means there are chances that the metaplasia can occur, and it can cause the carcinoma. And another, the strain of the H. pylori strain is exposed to many influence the risk of developing gastric cancer, because every H. pylori will not cause the gastric cancer. There are certain strains of H. pylori are more prone to develop the gastric cancers. The extract strains of the H. pylori that produce the high levels of two proteins, this is important, the vacuolating toxin A, you can detect it, and the cytotoxin associated gene A, that I have already mentioned it is the CAG-A, that is the island, appear to cause the greater tissue damage to those that produce lower levels, that lacks those genes completely. That these proteins are directly toxic to the cells lining and signal strongly to the immune system that invasion is underway. As a result of the bacterial presence, neutrophils and macrophages set up residence in the tissue to fight the bacterial assault. So the war is going on in the stomach when there is inflammation the acute inflammation of the stomach will causing the more damage. Now the H. pylori is damaging itself. It is responded by the body as the site as a chemotoxic phenomena, and then the acute inflammatory cells are invaded, where the wall is going on between the neutrophils, and then afterwards the lymphocytes, and damaging occurs and occur. Dyspepsia also is seen. Those people who have got only dyspepsia, that is called the non-ulcer dyspepsia. This is a research which is going on. So these dyspepsia occurs in about 20% infected individuals. So it may at one time it is called functional dyspepsia. Now that is a research, very new research said that the non-ulcer dyspepsia may be enhanced by the H. pylori. So this is a new research. By reduced protein expression of DNA repair proteins, MLH, MG, MT, MR, etc. Now these are the things which are important to understand, because in those people who have got dyspepsia, now dyspepsia means that it is the non-ulcer dyspepsia. Now survival of H. pylori, I already mentioned out that it is the survival which is important in the high acidic pH, or rather the optimized pH, it will survive. And the phagocytosis occurs, and accompanied by the release of the free radicals. These are important in the pathogenicity of formation. So this is the first part which I have now. It is actually a very dry type of subject, but most of the people, they don't want to listen. You have to listen, because the examiner, if he asks the microbiology, the pathophysiology, the pathogenicity, all things should be known to the students. Otherwise, he will reduce the marks. It means that you have not read properly the recent advances which have these are all recent advances. You don't know the recent advances. So 20% marks will be gone in the long case. 20% means 2 marks out of 10 is 0. So 8 marks are remaining. 8 marks mean that you have to fight for the 8 marks, for 7 at least, to pass this long case. Otherwise you will go. So that is why I am actually doing my best to provide you a recent knowledge, and this is first part which I have taken. The second part will be inshallah discussed very on the next day or day after tomorrow. If those who are interested can watch this video and can comment. وما علينا إلا البلاغ والسلام عليكم ورحمة الله وبركاته.